Bacterial protein overexpression? Just the gist version. Basically, inside of this flask are lots and lots of bacteria that I've turned into factories for making a protein I want to study. Now, normally the bacteria wouldn't want to make this protein, and they don't even have the instructions for making the protein. So I give them those instructions in the form of a circular piece of DNA called a plasmid, in which I put those instructions for making this protein. And then I stick that plasmid into the bacteria. It's a process called transformation. And so then these bacteria have these plasmids. But this bacteria still have no interest in making my protein, which is okay, because I want them to just grow and not worry about making my protein, just grow and get lots and lots of bacteria copies. And then once there's a lot of them, I want them to make my protein. We call this induction when we get them to make our protein. And so the way that I got them to make my protein is by adding a chemical called IPTG. So IPTG mimics like lactose. And so that's a type of sugar, and bacteria don't normally like to use lactose because they prefer to use glucose and things like this that are easier to break down, and they don't have to make special enzymes to process them. So instead, they only make lactose when there's not much glucose and when there's lactose present, because why make the machinery for breaking down the lactose if there's no lactose around? But when there is lactose around and they don't have that glucose that they really like, well, now they're going to start breaking down lactose. But in order to do that, they've got to make all those enzymes for breaking down the lactose. So they have a way to know if there's lactose present. There's this thing called the Operon, which is basically the set of genes that works together in order to make all the stuff that you need for lactose break down. And so in front, part of that Operon, like in front of it, controlling the whole thing, there's like this promoter and then like a repressor. Bottom line, they only make, when there's this lactose around, molecules kind of like unbind from the repressor and they bind to the promoter, and then the bacteria start making the protein. Well, those proteins for the lactose break down. Well, actually, they don't make those proteins. First, they have to actually make the messenger RNA for making those proteins. So the promoter is actually in front of the gene, is controlling like RNA polymerase going and making a messenger RNA copy of that DNA, and then that gets translated by the ribosomes into making a protein. And so in order to make the protein, you first need to make the mRNA, and in order to make the mRNA, you've got to have the bacteria be like, oh, okay, well, this promoter says go, and the repressor says, okay, no repression here, and so let's go and let's make those lactose making things. So now we've talked all this way about lactose, and now I'm going to tell you there's no lactose even involved in this story. Remember, we're using that lactose mimic, IPTG, because it's more stable and it's not going to like break down, and then it's, all that stuff. So we use this IPTG instead of lactose. Okay, that's great. We've got one thing, but we've still been talking about this Operon, and we don't care about that Operon. We just want our protein made, and our protein's not part of that Operon. So we trick the bacteria into thinking that it is. So we take that lac promoter, that thing that was in front of the Operon, in front of all those lactose breakdown genes. Instead of having them in front of the lactose breakdown genes, we stick that promoter in front of our gene of interest. And now what's going to happen was the bacteria, they're like, oh, there's lactose around. And there's not even lactose around. It's IPTG. But that fools them into thinking there's lactose around. And then having the lac promoter in front of our gene fools them into thinking that they're making something that they need to break down lactose, and then they really go and they make the messenger RNA for our protein. And then because there's a lot of that messenger RNA, the ribosomes are going to be like, hey, let's make some protein from that, and so you're going to get your protein made. And so that's the basics of when you do use a Operon to express a protein of interest. So express is really just kind of like a word we use for make, so we're having the bacteria make lots of this protein for us. In fact, we're overexpressing the protein, so we're getting the bacteria to make tons and tons of it. And they make so much of it that they kind of stop doing anything else. They stop growing, they stop dividing. They just focus most of their attention on making our protein. And so we get them to overexpress this protein, and we end up with lots of protein. But first, we got to let them grow. And so that's why we induce the expression later on. We don't just have them make the protein from the beginning, because instead we want them to devote their energy to duplicating, so replicating, making lots of copies of themselves. And then when there's lots of copies of them, then we induce that expression. And so we monitor the growth of the bacteria by measuring something called the optical density, or OD. Basically, the more bacteria there are in the solution, the less light can go through. And so if we measure how much light goes through, then we're going to be able to measure how much bacteria there is there based on the bacterial cells kind of like scattering the light. So we measure the OD. We find when they're in this kind of like phase where they're doubling and doubling and doubling, and but before they run out of nutrients and like plateau and then start dying. So we find that sweet spot, like that mid-exponential range, and then we add the IPTG and convert these into protein-making factories. And so I did that, and so hopefully in the morning I will see that the bacteria have made lots of my protein. In fact, these bacteria I was way too ambitious, and I made a liter of media, but then the shaker would only hold up to a 500 ml flask, but then the screws didn't work for that. So now I'm doing 500 mils in 4 125 ml flasks. So yeah, you get to be really creative when you're working at a PUI, which is actually pretty fun. So yeah, if you want more details about the overexpression, I have much more on my blog and in the figures. And I have a longer video on it as well. Note that sometimes we have the promoter in front of the T7 polymerase, and then we have a T7 polymerase promoter in front of our gene of interest. That's kind of like taking things to another level. You see, if you see like DE3 cells, they have this sort of system set up, like PET 28 plasmids and things like this. Basically, this is a way in which the lac operon, when you add the IPTG, it tells the cells to make the protein called or T7 polymerase, and that polymerase is one of those mRNA makers, but it's not the same one that the bacteria use for their own stuff. And so we get them to dedicate to all of that new one that we have them make to making a protein that we want by sticking the T7 promoter in front of our protein, gene for the protein of interest, but it's not in front of anything else because it's this phage promoter. So it's basically this bacterial virus. And so the bacteria don't normally have this stuff, that promoter, in front of their own genes. And so by putting it in front of the gene that we want, we're then able to use the T7 polymerase instead of the bacterial polymerase, and so we don't have to compete with that bacterial one. So we get them to make even more of it. And by having that like 2-layer security system, you can kind of prevent some leaky expression where things just kind of get made before you induce them. And so that's another way that you can use this lac operon is through having the lac promoter control the T7 polymerase, and then the T7 polymerase promoter control your gene. And sometimes you get like a combination where you actually have like a T7 promoter and a lac repressor and promoter in front of your gene of interest. And so you can get extra security to make sure that you're not getting things made when you don't want them to be made, like if your protein's toxic to the bacteria or something like that. So again, much more in other posts. This is just the gist.
Video này do mình, Cương bên npslab.vn, tự chỉnh sửa phụ đề hoặc lồng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.